Again. Let's do it quickly again when we have sound.
Of APC and helper T cells, it recognizes the MHC1 complexes on the infected body cell's surface. Cytotoxic T cells then release two molecules, perforin and granzyme, in order to destroy the infected body cells and the pathogens that infect them. Perforin creates pores in the target cell membrane. The pores allow extracellular fluid to enter the cell, causing it to swell and potentially rupture the cell membrane. Granzyme then enters through the pores and causes the degradation of its proteins to ensure cell death. Cloned B cells do not attack individual cells. Instead, after they become plasma cells, they greatly increase their production of antibodies. These antibodies are released into the plasma and extracellular fluid, where they will match and bind to the target antigen. Antibodies are Y-shaped structures with unique antigen-binding sites on the tips of two of the arms. This allows an antibody to perform different functions depending on the type of binding site it possesses. They may essentially prevent invaders from interacting with normal body cells, or mark foreign cells for destruction, and activate our defensive cells into specific action.
Okay, that brings us back up to speed. We had talked about antigens and epitopes, how we target and recognize an invading pathogen, and these were examples of the pathways by which we fight off those invading pathogens. Cell-mediated and antibody-mediated immunity. And we'll do a little elaboration on those details. Not in depth. There's more detail in the book if you're curious, but we are just talking about each of these components at a relatively high level. Before summarizing, quick lecture, but we're going to do some fun stuff after lecture today. So, cell-mediated immunity, carried out by cytotoxic T cells, is where an antigen is recognized by T cells through presentation at the cell surface. And T cells will either register that antigen in infected cells, that's endogenous antibody processing, we talked about that last class, or they can recognize it on APCs, antigen presenting cells, where the antigen was floating around free, the APC engulfed it, recognized that it wasn't supposed to be there, and now is signaling the T cells to induce this branch of the immune system, the cell-mediated immunity branch of the immune system. So T cells are central to this arm. If they recognize the antigen, they will want to mount an immune response, but they can't do it alone. They need to be what's called co-stimulated or helped. If they are co-stimulated by helper T cells, then clonal selection will ensue. And we know clonal selection, it's when we take a T cell and it divides into a new T cell, a new cytotoxic T cell, and a new memory cell. So we have an effector cell, which will do the job of the immune system, the cytotoxic T cell, and we have a memory cell for later that remembers that it divided. So if the T cells are co-stimulated, clonal selection ensues. There are more cells available to be involved in this cell-mediated branch of the immune response. The T cells will recognize either of the antigens. Helper T cells themselves are activated by those dendritic cells, the APCs specifically. Now we have T cells available, clonal selections ensued, the new active T cells then travel to the sites of infection, wherever they happen to be. When they travel to the sites of infection, they kill the antigen. They kill the microbes, they eliminate the cells through the mechanisms that we saw in the video and the mechanisms I will show you briefly on the next slide. So T cells are induced. They are co-stimulated by helper T cells. They travel to the site of infection. And they destroy the antigen as follows. So the first kind of cell targeting or cell destruction is through the actions of granzyme. Granzyme is a special protein that induces cell death, that induces apoptosis. That's the fancy science word for cell death. So if a cell has been infected and it's recognized by a cytotoxic T cell, granzyme will make that cell burst. It will make the proteins unravel. The cell will become non-functional and will die. In bursting, it releases the invading microbes that will themselves be destroyed by phagocytosis. So granzyme is one arm of this cell destruction pathway instituted by cytotoxic T cells. The other branch, and I don't know if the names were slightly confused, it's the same principle as we saw in the video. The other branch is through a protein called perforin that pokes holes in the membrane of the infected cell. Water and fluid rushes in, the cell enlarges and swells and will eventually burst. But those holes also allow another protein to enter that does something very similar to granzyme. The name is also very similar to granzyme, called granulysin, which causes lysis or rupture of the granule of the cell. So very similar approaches. Granzyme alone triggers apoptosis. Perforin allows granulysin in to do the dirty work and trigger apoptosis, break down the microbes, and cause cell death. So if infected cells are registered by cytotoxic T cells, these are how those cells are destroyed. But infected cells, invading pathogens, aren't only recognized by cell-mediated immunity, they're also recognized and acted upon by antibody-mediated immunity. And how antibody-mediated immunity is induced, we know that B cells are the primary cells involved in this response, but they need to be activated. They are there and they're present, but unless they are activated, they won't do anything. They won't release new antibodies. And these dormant B cells have receptors on their surfaces that will bind a wide variety of antigens. And those receptors that are bound will wake up the B cells. When those B cells wake up, that's when the antibody-mediated arm of the immune response kicks into gear. The receptors wake up the B cells. Like we saw on the cell-mediated side, these cells want to mount an immune response, but they can't do it alone. They need secretions from helper T cells to induce clonal selection. Very similar pathway here as on the last slide. They wake up, but they need more B cells to mount a larger immune response, and the way that clonal selection is induced is if helper T cells are present to co-stimulate. Clonal selection again makes new effector cells and new memory cells. In this case, the effector cells are plasma cells, and the memory cells are memory cells. The plasma cells are what secrete antibodies into the blood. And this is an important point. The antibodies that are released are specific to the original signal. So the invading microbe, with its epitopes, will bind only specific receptors. So it will wake up only specific B cells. And as those B cells are co-stimulated and differentiate through clonal selection, they'll start to produce antibodies that are specific to the original trigger, which is good. We want these new antibodies to bind to and target the original microbe. So because the receptors responded originally to the microbe, the antibodies that are produced later will seek out and bind that same microbe. And they bind it, they inactivate it, or they flag that microbe for destruction by other helper cells in the immune system. Now, these are the two main mechanisms or pathways through which the immune response is mounted, and one of the major features, the hallmark features of the immune response, is that it has a memory that allows it to ramp up faster and to a greater degree over time. So what we're looking at here is an example of an antibody-mediated response to a given pathogen. And the antibody titer is a measure of antibodies in the blood. Just how much is in the blood. The more there is, the more of a response is being mounted. Also note, antibody is immunoglobulin. Those two words are interchangeable. They mean the same thing. These traces are immunoglobulin G and immunoglobulin M. There's different kinds or classes of immunoglobulins. That's not important. Immunoglobulins are antibodies. So on the first response, there are few cells with the specific receptors to respond. And so we have a small blip in the production of antibodies as shown on the left-hand side. But after clonal selection, and with this now larger population of memory cells, more cells have the receptors that can respond to that antigen. More cells can undergo clonal selection and produce antibodies. And therefore, we have a much larger and faster immune response. On the second and subsequent exposures, the immune response only gets faster, ramps up quicker, and it's more specific. The antibodies that are produced in this second exposure actually bind tighter to the invading microbes. Quite the adaptive response. This is the basis for immunization against certain diseases. This is how we can boost our immune system, so to speak. Vaccines are administration of weakened microbes that will cause an initial response that allows a greater pool of B cells and T cells for future invasions. In fact, there are 4 ways that we can enhance our immunity or develop acquired immunity. The first one is natural active immunity, and this is what we've been discussing in this section. You get sick, you fight it off, you have new B and T cells in place that can mount an immune response in future. Natural active immunity. Another way that we can enhance our acquired immunity, boost our immune system, is natural passive immunity. Here, antibodies are delivered to the host, delivered to the person. We gain these from an outside source, and that outside source is often from the mother to the child through breast milk, through the birth canal, through childbirth. These are immune features and antibodies that the child wasn't producing initially, that were given them by the mother, that enhances their immune system, improves their acquired immunity. Both of these are natural ways that our immune system is boosted. There are artificial ways that we've developed to boost our immunity as well. We can induce an immune response artificially. One of the greatest advancements of modern science. We can deliver inactivated or weakened microbes that cause B cells and T cells to proliferate, increase the pool of memory cells to enhance our immune response to a later invasion. This describes the process of vaccination. That's why polio doesn't run rampant nowadays. Or you can infuse antibodies intravenously. An artificial passive form of boosting your immune system could be to deliver antibodies directly rather than stimulating their production by infusing them intravenously into the blood. 4 ways through which we can boost our immune system and enhance the response to future invasions. I want to summarize. This is very quick, but I need to stay on time. I would bring up the intro slide to tell you the purpose of the section, but we covered all the points, not to worry. Summary. What did we cover in this section? Lymphocytes are major soldiers in the battle against invading microbes, invading pathogens. Lymphocytes are the major white blood cells that mount an immune response. B cells, T cells. We didn't speak much about natural killer cells, but they are generally broad killer cells that can destroy a wide range of cells. Innate immunity was the immunity that is non-specific, a general physical barrier or enzymes that target many pathogens in a broad sense to protect against invasion. Second aspect of our immune system was adaptive immunity, which we just finished discussing, where specific responses are tailored to an invading antigen or pathogen, and where a memory is created whereby future responses are faster larger, and more specific. And lastly, the immune response overall isn't one or the other. It is both cell-mediated and antibody-mediated. Both arms will turn on depending on where the antigen is found within the body. If cells have been infected, maybe you have a larger proportion or a contribution from cell-mediated immunity. If few cells have been infected, maybe a larger contribution from antibody-mediated immunity. But both will respond. It's not one or the other. Any questions?